All right. Well, luckily, this part that we just finished, is not new, right? We have already discussed this in previous instances. So let's start from here. Yeah, so all chordates are triploblastic, right? Bilaterally symmetrical coelomic. So they carry those deuterostomic features into the chordate clade. Now, those are not unique to chordates, right? And sexes are largely separate. Um, Fertilization happens both externally as well as internally, right? Especially in non-vertebrate chordates, the fertilization is external, right? They release their gametes to the water, and in water, fertilization happens, right? And lots of chordates still have larval stages. Many vertebrate chordate lineages have lost the larval stage, right, or have shortened the larval stage, but many non-vertebrate chordates actually have larval stages, and there are lots of interesting things we can talk about their larval stages down the road. um so now, what are the unique characteristics to chordates? Not the carryover ancestral features, but what are the actual synapomorphic features? Notochord, right? Where do you find notochord, and what's the purpose of that notochord? The characteristic of the notochord, it's flexible, right? It's a rod located between the digestive tract, above the digestive system, um, and the nerve, and below the nerve cord, right? That's kind of where notochord is located. Its function is actually to maintain the body shape, right? Provide surfaces for muscle attachment to support the body, right? Um, then the noto, and then the secondary feature is dorsal hollow nerve cord, right? It's dorsal to the notochord. It's kind of like on the top of the body, right? And then its function is to conduct nerve impulses, right? It's a part of the nervous system. Pharyngeal gill slits. Um, so pharyngeal gill slits, they are located, observed externally. It's kind of, you can observe the slits from the outer surface of the pharynx, and their job is actually gaseous exchange, right? Of course, it's a gill slit, so it does help gaseous exchange, but that is not the only function, right? In certain chordates, it has become so elaborative simply to support suspension feeding, right? To do filter feeding, to trap suspended food particles. Last, post-anal tail, right? So mentioning it is post-anal tail is extremely important because there are other invertebrates that are not chordates that actually has a tail, like shrimps, right? Like nematodes. But this post-anal, meaning the tail occurs after the anal pore, right? Um, and it is located posterior to the anal opening, right? What is its job? Well, it is job A, to keep the balance. Now, that's something you would observe in many other tails you observe in non-chordates. But post-anal tail can also propulse the animal, right? Create the propulsion force when animal is swimming. So now the question is, okay, those are four critical characteristics of chordates. But what happens to those chordate features in many derived vertebrates like primates, like humans? Notochord disappeared almost entirely except for those discs that the cartilaginous discs that occur in between vertebrae, right? So now that cartilaginous discs you find in between vertebrae in the vertebral column collectively makes the vertebral column. So it's not incorrect to say notochord helps to make certain parts of the vertebral column. That's not incorrect at all. But do not assume that the notochord modifies into the vertebral column, because the actual vertebrae in the vertebral column are not coming from the notochord, right? They actually develop subsequently to the residence of the notochord, right? Whatever the receded parts of the notochord remains as gelatinous cartilaginous discs between vertebrae. It's really important to understand that. Dorsal hollow nerve cord, well, that's easy. It becomes spinal cord, right? And the pharyngeal slits or pharyngeal gill slits or pharyngeal gill foldings in humans and primates become part of the inner ear, right? And the neck region. And the post-anal tail, well, completely resides in humans except for the coccyx, right? Uh, you can say the same thing regarding apes, right? What is the biggest difference between apes and other monkeys? Anyone wants to take a stab at that?
They don't have a tail.
Correct, correct. They don't have a tail, right? So for all of them, actually, the post-anal tail shortens dramatically until and end up becomes just the coccyx. You know what the coccyx is, right? It's actually at the end of the vertebral column. Kind of it joins the pelvis. Any of you have any bad experience breaking the coccyx, right? So now let's now move on to any questions regarding chordates. Now these parts may have been already covered in your lecture. In my lecture part, I haven't covered those yet, but alrighty, let's move on. Echinoderm characteristics. Well, echinoderms are exclusively marine, right? There are no freshwater forms, there are no terrestrial forms, exclusively marine, right? So echino means spiny skin, right? And also they have something called ossicles. And let me actually show this particular figure over here to explain what an ossicle is. Ossicles can actually undergo many different modifications or have many different formations in the echinoderm lineage. So in this one, this is a sea urchin. In them, the ossicles completely and permanently fuse to each other, right? And make a very holistic, rigid um, skeletal structure, right? If you zoom into this particular thing, first you see something like this, right? Particularly if you do an scanning electron micrograph, it actually looks like this, right? And then if you like zoom into a very tiny part of that, it looks like that. So here you can see actually there are, you don't even see the margins in between ossicles because ossicles have completely fused. So ossicle, imagine yourself as a very small skeletal element, bone-like components, right, that can remain individually separately, right, or fuse completely, right? And these guys are triploblastic. They are deuterostomes, have a coelom, um, and adults are pentaradial symmetrical, right? Adults have pentaradial symmetry. Larvae have bilateral symmetry. Now, if you wondered, you should, going back at the lineage, right? Bilateral symmetry evolved way back in that lineage, right? So how did they actually complete? If if the adults have pentaradial symmetry, how can you include them in a lineage where the common ancestor were bilaterally symmetrical? Well, the answer to that question is, well, the larvae still has bilateral symmetry. What happens is actually that bilateral symmetry amplifies into pentaradial symmetry in the adult, right? It is something happened in the developmental process in echinoderms, right? So it is actually, so don't mix up these guys as the radial symmetrical creatures, organisms we talked before, like coelentrates were radial symmetrical. These guys, phylogenetic relationship with cnidarians or coelentrates are quite deep, right? These guys are much more closely related to us than they are to other radially symmetrical invertebrates. So keep that in mind. They are not closely related to other radially symmetrical animals, period. Water vascular system is actually a really interesting organ system in their body. Actually, I should show you a different diagram to describe what that is. So generally what that is actually vascular is a tube system, right? Water transporting tube system, right? It's what a water vascular system is. It actually occupies the entire not the body volume, the water vascular system spreads throughout the entire animal, right? So it is composed of vessels, vascular systems that are canals, right? And then those canals ends up in tube feet. So the whole purpose of the water vascular system is to create a hydraulic force, right, inside the water vascular system that triggers, that helps movements of different body parts as well as of the entire animal. Um, they actually have an internal skeleton, right? This particular ossicles, that actually fuse together is not external, it's rather internal, right? And so these ossicles are actually, another term for these ossicles are actually bony plates. That's what simply an ossicle is, right? It's a bony plate. The size of those bony plates could be large, like can be observed to your naked eye, like in a starfish, or could be microscopically small, like in a sea cucumber. Um, they do have complex organs and organ systems, like a reproductive system. And interestingly enough, in mature echinoderms, like mature starfish, during the reproductive season, the gonads grow so much, they might actually, when you dissect a very mature starfish, their limbs could be entirely filled with humongous gonads, right? Um, in terms of their lifestyle, it's extremely diverse. Starfish are predators, right? And some echinoderms are scavengers, right? And then certain echinoderms like sea lilies are filter feeders, right? So you have a huge variety of live feeding styles among echinoderms. Um, their sexes are separate, right? Male and female separate. They actually release their sperms and eggs to water, and fertilization happens externally. Larvae, and then they create a free swimming larvae, which actually undergo development to produce an adult eventually. Very few echinoderms are mobile, are sessile, like sea lilies. I will show you some examples to them. And most of echinoderms are sessile. They can move either quite fast, right, or slowly, right, or creep, right, very slowly. And some of them can move really fast. Um, if we were to take a look at a couple of these pictures, anyone, anyone wants to guess what these things are? This is a starfish limb you are looking at. Anybody want to make a guess what these things are?
Tube feet.
They are tube feet, Emily, correct, right? So in fact, tube feeds help them to A, primarily to move, B, secondarily to capture food, right? So these tube feeds are quite flexible. They can be extended and retracted back, right? Well, how and why? How and why? Well, remember the water vascular system I talked about? Those tube feeds are connected to the water vascular system inside the limb, right? So, in fact, part of the water vascular system. So, water can be pumped in and out of the tube feed. By that, you can either extend or retract the tube feed. When you pump in, it can be extended. When you take the water back from the tube feed, it retracts, right? So that's actually how hydraulic force is created to move the tube feeds. Tube feeds, you may ask, okay, can they individually manipulate each tube feed? Yes, they might have thousands of tube feeds in them, but they can actually individually manipulate each and every tube feed. Can they coordinately manipulate all the tube feeds in a single limb? Yes. Can they coordinate like a part of a set of tube feet within one limb? Yes. So they actually, the maneuverability of those tube feeds are amazing. All right. Anyone has a question? I thought somebody was. I heard some mic noise. Any questions? Nada. So, and over here we are looking at the internal anatomy, right? Let me explain the internal anatomy and get a little bit into the water vascular system as well. Um, so the water vascular system should require water, right? So how do they get water? Well, not from the mouth, right? If you look at the top of the body, right, there is an opening called madreporite, right? That actually sort of is the opening through which these animals get water. So madreporite is actually an opening, right? It's a hole, right? It's not the same as the mouth, right? It's not the same as the anus. It's a separate opening on the in the center of this animal, right? So if this is the center, which is also called the central disc. So to the side of the center, right, you actually you locate this madripoor, right? It's a big visible structure, right? And this particular structure is actually hard. It's not soft because it's made out of calcium. And then from the madreporite, it actually connects to this particular thing called ring canal, right? And then ring canal radiates into each arm, right? And that radiating canal is called radial canal, right? And the radial canal creates, in each limb, a single radial canal creates multiple lateral branches called lateral canals, right? And each lateral canal connects to a tube feed. Tube feed actually has an ampulla on the top, which actually can squeeze water in and out to the podium, which is actually the part that is externally visible that can either attach to a substrate or detach from the substrate, can attach to food particles, detach from the food particles. Okay, um, that's actually good enough. It's really important to understand, be able to label the parts of the hydrovascular system, right? If it is not clear, you need to ask. So what other unique characters do you see in echinoderms? First, let's take one picture at a time over here. So this is actually what the surface of an echinoderm looks like, right? You see lots of these kind of large spines, and you are looking from above onto the skin, so you don't see a spine um, like this, right? This is actually how a spine looks like from the side, but this is how a spine looks like from the top, right? And amongst the spines, you are going to see this kind of a reddish, pinkish colored, very small, tiny structures, a lot of them, right? Like all over the skin, right? Now, um, some of the structures, if you actually like pick up a tiny fragment out of that and put it under a light microscope, right, you are going to see things like this, right? So these parts that are extremely small, right, spine-like but much smaller than an actual spine, right, are called dermal branchiae, right? Anyone wants to make a guess on what dermal branchiae does? So what is a dermal branchiae? It's actually a protrusion, very small, microscopically small protrusion on the surface of the skin, right? And they are, you can say they are very flimsy, they are very delicate. And in this particular photograph, they seem to be transparent. Anyone want to make a guess what they do? Think about the word brachiae. Respiration, right? They actually have, they do perform gaseous exchange. And then, so these are the dermal branchiae, and these are actually pedicellaria, right? Pedicellaria is actually, diagrammatically looks like this, right? And or like microscopically, they either look like this, but if you zoom further into those, they look like that. So you clearly see two jaws, right, in each pedicellaria, and the pedicellaria has these jaws, and then of course a stalk. So because of the stalks, the pedicellaria can be moved, right? And the jaws are also mobile, right? As you see in this diagram, right? These jaws have muscles. Now, I do not expect you to memorize this particular diagram. It is just to teach you like those jaws can be movable, right? Um, and another diagrammatic picture of a skin is over here, right? You can see spines over there. They are more rigid, hard structures, that actually are pointy in their end, and the dermal branchiae, right, are actually, are more blunt, right, protrusions of the skin, right, that where sort of the skin surface protrudes externally, right. And in between dermal branchiae, what you get is those ossicles, right, those bony plates that collectively makes up the skeleton, right, internal skeleton. As you can see in this picture, these ossicles are isolated. They don't fuse together, right? They might hinge, but they are not fused particularly. Now, let's move on. Um, Now, there are several classes in echinoderms, right? You have asteroids that actually includes sea stars and sea daisies. This is a sea daisy, right? Daisy, it's called a sea daisy because it looks like a flower, right? And sea stars, you all know what a sea star looks like. Um, and then these guys are largely they are predators, right? Actually extremely important predators in coastal ecosystems. If you remove them, you actually even lose the biodiversity in coastal ecosystems. um, they can slowly move across harder surfaces. They have a hard time moving across softer substrates, so they largely occur in rocky substrates in the intertidal zone, or in much more harder surfaces in coastal environments. Um, in these guys, they are ossicles, the bony plates, they do overlap, right? They don't fuse necessarily, they actually overlap. Um, and then we get to the class Ophiuroideae, where you get basket stars. This is a really weird animal, like looks like over here. And we have brittle stars like this. Now, there's a reason why we call them brittle stars, right? Because they are literally brittle, right? They actually can brittle. They can, if you handle a brittle star very roughly, if you handle it very carelessly, you might break their limbs, right? Guess what? They will regenerate the limb they broke, right? They're pretty good at that. Um, so they are also predators, right? They in contrast to sea stars can move much faster, right? And some of them can actually swim. Um, their ossicles don't overlap, rather they abut. So because of that, they actually have a hinge between their ossicles. So because of the hinge, they can move um, their limbs. Their limbs are much more flexible, right? Like a door, they can move their limbs. Um, the hinge between their two ossicles can move like a door, right? So because of that, their limbs are a lot more flexible than the limbs of sea stars. Okay, Holothroids, sea cucumbers, right? Anyone wants to mention an economic importance of sea cucumbers? Let me ask a question. Any of you have sea cucumbers as your poster animal? I actually honestly don't remember. I don't know whether they are raw. So the point I'm bringing it up is, can someone tell me an economic importance of sea cucumbers? Anyone? You can eat them, right? People actually eat them. There are sometimes in certain areas, the consumption is so high, certain populations are declining in certain coastal regions. So, and basically, the sea cucumber looks like this, right? Or some of, most of them are actually just looks like, a piece of poop. Honestly, these are called donkey dung sea cucumber because they actually look like poop of donkeys, right? And what they have is actually, on the cephalic, the anterior end or the cephalic end, they have lots of tentacles. The shape and the morphology of those tentacles can be highly variable. Look at in this guy, those tentacles looks like mushrooms. In this guy, the tentacles looks like a tree, right? So um now these guys actually don't have large prominent ossicles. What they actually have is their ossicles have become extremely reduced, right, and become microscopic, right? In certain individuals, they have completely lost all ossicles. So they actually have ossicles, but during the development, they actually have become extremely smaller, microscopically smaller, or have completely lost them. Um, Predatory, right? And some of them actually are known to be filter feeding, but largely they are predatory. The mouth points forward, and what they also do is they can eject their entire digestive system as a anti-predatory mechanism, right? As a defensive maneuver. They literally vomit the entire digestive system externally. Now, here's a question for you. What do they do after that? right? Do you think they can actually take it back inside?
Yeah, I think they do.
Okay, remember what I mentioned about their amazing ability to regenerate? What they do is they don't necessarily take it back in, they let it go and regenerate their entire digestive system from the scratch, right? Imagine what happens when you like vomit your entire system out. You could get damaged, right? Like those structures are extremely flimsy. They don't have like like very strong muscles around them like we do, right? So what they do is they will like let it go and they will grow it back. Really fascinating creatures. So speaking of which, another economical importance of any echinoderm is medical research, right? You know, unfortunately, if we our limbs had to be amputated, we cannot regrow them, right? We have a very limited capacity even to regrow our nerves if damaged, right? Things like our regeneration capacity are extremely limited. Liver can regenerate, but sometimes it doesn't regenerate properly. Heart muscles cannot regenerate properly, right? Lung can regenerate to a certain degree, right? Muscles can regenerate to a certain degree. Bones can regenerate to a very substantial degree, but most of our body systems cannot regenerate well. So these animals, no matter where they lose their limbs, what organ they lose, they can regenerate. There's only one documented, scientifically validated incident when that they lose their regenerating capacity completely. There's a virus, a wasting viral disease they get. If they get it, they completely lose their regenerating capacity. So what happens is when infected starfish that contracts this regenerating disease, limbs fall off as you watch, and they never regrow them back. They die subsequently. But other than that, these guys can regenerate in an amazing level. Sea urchin echinoderms is the example for sea urchins are the examples for echinoderms. There are other examples within that particular class. Sea biscuits, sand dollars. So sea urchins are actually sort of like a compressed globe, right? Um, like this, right? And some of them actually retain their globular shape like this, right? Some sea urchins can be somewhat compressed, but sea biscuits and sea dollars actually are completely compressed, so they are flat, they are disc shaped, right? Um, Echinoderms in generally are herbivores, right? They are grazers. They literally graze. They have teeth to graze on algae, right? And they can also move across harder surfaces because they have tube feet that they can extend from their body. Um, and unlike starfish, echinoderms can maneuver themselves even on softer surfaces. Even on like sandy surfaces. In fact, sand dollars are so called sand dollars because they embed themselves into the sand, right? Um, and if they don't like that particular place, they can actually even dislodge themselves and move somewhere else on the sandy surfaces. Um, they also have a really, really cool structure called Aristotle lantern, which is located interiorly, right? Which they use for macerating food, um, breaking down the food they take in. So Aristotle lantern is actually a huge internal structure that are found on these creatures, and I will actually upload a particular link with nice photograph to those Aristotle lanterns. Um, that is comprised of calcareous, like calcium carbonate made structures that and muscles. So muscles move, can contract and move these calcareous calcium carbonate structures around, and they can collectively sort of macerate the stuff they eat. It's really important that they do so because if you are eating vegetable, you need to break down the cellulose, right, to take up the nutrients. Secondarily, some of those sea urchins actually ingest sand or rock particles and use the Aristotle lantern to grind the rock and sort of scrape the algae or algal films growing on those particles. Um, have you heard the fact that echinoderms are poisonous? Yes, those spines in them, right? If you actually, if you skin get pierced by those spines, you might actually have a very serious medical problem, right? It could lead to um, in certain cases, like it could even lead to a massive tissue damage in your limbs, particularly if you walk barefooted in rocky intertidal environments or shallow coastal environments. This could be a problem, right? And those spines are extremely sharp, right? Much sharper than the spines on a starfish. And generally, in most echinoderms, particularly those who can move around, their mouth is not on the top, the mouth is actually on the bottom, right? And of course, like I said earlier, their ossicles fuse, making a very hard structure, right? And that particular hard structure is not external, not an exoskeleton, it is an endoskeleton, right? So what you see in a museum like these, sand dollars or like urchins, that's actually preserved exoskeleton after the animal actually dies. In live specimen, it looks more like this, right? And again, even in like in both starfish and sand dollars, you can see the pentaradial pattern, right, over here, like 5 petals of a flower, like in this case, right? And over here, 1, 2, 3, 4, 5. So the pentaradial symmetry is actually intact. Crinoidea is a really interesting group of echinoderms. They are, by the way, the most ancient lineage of echinoderms, and they are most of them are sessile, right? Meaning they anchor themselves to the ocean floor and stay there, right? The larval stage is the only mobile form that they get. Sea lilies and sea feathers are the examples. They're extremely beautiful. They look like a seaweed, right? But they're not, right? They actually resemble a tree, right? Look at this one. Like, it doesn't look like an animal at all. None of them look like animals. Um, their oldest group of echinoderms, oldest group of extant, currently living echinoderms. Um, these guys, unlike any other, are filter feeders. Their mouth points upward, right? Like, these are their arms, the radiating arms, and the mouth is located right in the center, right? So unlike other echinoderms who had their mouth on the bottom, like starfish, like sea urchins had their mouths on the bottom, these guys have their mouth on the top. They have um, in this guy you actually see 1, 2, 3, 4, 5, 6, 7, 8, 9, about 10 arms, right? And other groups of crinoids may actually have multiples of 5, right? I cannot think about any of them having just 5 arms. Most of them actually have multiples of 5 of arms radiating from their center. Some of them actually have a long stalk, right, between the arms and their anchor, like this guy, and some of them may only have a very short stalk, right? Um, um, So in sea feathers, it's kind of interesting. In juvenile sea feathers, they usually may actually have a stalk and they might attach themselves to the substrate, but as an adult, they could dislodge themselves from the substrate and swim, right? But sea lilies, in contrast, they are the larvae are free swimming and adults are anchored permanently, right? So sea feather are the only exception where the adults can actually dislodge from the substrate and can swim. All right. We will go to the exercise later. So let's talk a little bit more about chordates. Before that, any questions on the sea urchin part? Or so rather the echinoderm part. Alrighty, let's move on. So we are going to talk about cephalochordates and urochordates as examples, example subphylum within the phylum Chordata. We will not talk about vertebrates today. So, right, what happens to, so let's revisit about these 4 chordate characters and what they do, right, in these animals, right? So notochord, dorsal hollow nerve cord, pharyngeal gill slits, and posternal tail, right? And then we already went over this. So what I want you to do is start thinking about what is happening to these four features in those two subphylum within the chordates, right? Um, so we did talk about that already. Um, Now in subphylum Chordata, right, the example is Amphioxus, right? Um, or Brachiostoma is actually one of the general genus names that we use, right? They are fish-like creatures, right? Very small. Look at this one. There are thousands of Amphioxus on your arm. Like they are not long, not more than an inch and a half long, right? Mostly like could be less than an inch in their length, right? Very tiny, right? Move like a fish, but the swimming motions are not as elaborative as a fish, but they can swim around, right? And mostly do they swim around water? While they can swim around water, mostly they don't. Mostly what they do is they anchor from their tail end onto the substrate and keep their mouths open like this and filter feed, right? So they, I would not call them sessile, but they can anchor themselves. And if they don't like where they are, they can easily dislodge, move somewhere else and anchor. Um, they can actually anchor way down into the substrate. Look at this guy, only about like 1/5 of the body is exposed. The rest of the body is anchored in, right? If you zoom into the mouth or the front end, you see something like this. This is actually the mouth, right? And you can see the notochord very clearly over here. And this is the pharyngeal region, right? So, um, cephalopodcoded characteristics. Now, when I explain this, I always want you to ask the question, what happened to the key coded features, right? So these guys, right, the cephalochordates, the Amphioxus group, they maintain all the four key coded characteristics, both as an adult as well as a larvae, right? All of them are like observable very clearly. Filter feeders, by their feeding habit, spend their life with their tail end embedded, anchored to the sand, right? And with the mouth pointing upward where they do the filter feeding, right? So they use their gills, right, in the pharyngeal region to trap, not only for respiration, but to trap food particles, right? And coelomate, they're triploblastic, but those are not just unique to cephalochordates. The other group is urochordate. It's completely different. They don't even look like a chordate. Particularly the adults don't show most of those chordate features, right? If they show it, it's extremely receded, extremely reduced. Um, so this this is Siona intestinalis, right? This is actually most of the urochordates are actually sedentary, permanently anchored to a substrate, right? And they, I can fool you and make you think that this is a sponge, or make you think that this is a nidarian, right? It looks like that. And most of those sedentary forms of urochordates are actually colonial. Look at this example, right? For instance, right? Some of them could be solitary, like this guy over here, right? The sizes are extremely variable, right? It could be quite small, as you can see in this diagram, right? Now, are all urochordates permanently anchored to a substrate? No. Look at this guy, a colonial urochordate, right? That is actually free swimming, right? It is not attached. It can swim as an entire colony. It looks like a chain, right? And chain comprised of multiple individuals, and they could actually, when they make a colony, they could actually share food, right, among different individuals, right? And in fact, there are certain free swimming or free floating non-attached urochordates that are extremely large. They are so big, and they make a tube, a complete tube. They can swim through that particular tube. In my lecture section, actually, I have a couple of videos on that. You can watch them. So yeah, so the sizes can be extremely variable. Well, now what happens to their chordate features? As a larvae, right? These urochordates will show all four chordate characteristics very clearly, right? But when they become adults, they undergo massive transformation, right? A huge change in their body form and function and style. Uh, they will recede all the characters except pharyngeal gill slits. And the pharyngeal gills, the pharynx in adults become much larger, occupies the entire space of the body, right? And so large you can actually visibly see them to your naked eye. They are coelomate, they are triploblastic, they are filter feeders, right? They actually use one siphon to draw water in and another siphon to expel water out, right? And that siphoning process helps filter feeding as well as respiration. Larvae can swim, they are free swimming. They look like a tadpole, right? But adults mostly are sessile. They have a hold fast, which actually sort of acts like a cement to attach permanently onto the substrate. Well, that is the end of the background. Any questions? Very good. Let's move on to the exercise, guys. Here we go. Da, 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 right. Still, are you looking at my slide? Invertebrate deuterostomes exercise. Very good. Very good. Again, make sure how you do this. You read the lab manual while you do the activity, right? And any questions embedded in your lab manual, you answer them on your lab notebook. First, let's do the key, right? What do you guys think? Are you by this stage, are you comfortable to do it on your own, or shall I go over at least a couple of examples? Go over a couple of examples. Give me a nod or a yay. Okay, I have nods and yes. Let's pick one of these right over here. Okay, take a look at the animal, right? You already can guess what this is. Now, I do want you guys to answer, so you may want to turn the mic on for this particular presentation. Um, Animal is bilaterally symmetrical. Animal is radially symmetrical. 1A or 1B? Give me a shout.
1B.
Animal is radially symmetrical. We go to 3. Body has 5 arms. Body is divided into 5 parts without apparent arms. Let me go back to the, what do you think? 2A or 2B? Sorry.
A.
No, I went to the wrong one, didn't I? So 3A or 3B? What is it?
3A. 3A.
Yeah, I hear many people saying 3A. Body has 5 arms, so go to 4. Central disc, very round, arms narrow, little towards the tip, meaning tapering arms, right? B is actually central disc slopes to arms, arms tapered towards their entire length. So first, central disc very round, right? Perfectly round, clear, separate central disc. Arms narrow, very slender, spaghetti-like arms, which actually extremely like tiny towards the tip. B, central disc sort of slopes and tapers towards the arms. What do you guys think? For A or for B?
Mm. A. For A.
For A, yeah, correct. For A, right? I mean, let me take my pointer back over here, right? So this is the central disk, right? You can clearly see the central disk, and it looks like, these very tiny, slender arms are sort of attached to the central disk, right? And the tips, they are extremely tiny, right? Yep, good. Let's go to this guy. Take a look at that guy. It says animal is radially symmetrical and soft bodied. Since you cannot touch it, we mentioned that. They are radially symmetrical. Yeah, and yeah, it's very easy if you actually speak up, right? Because it's harder for me to keep checking the chat. Let's go for a second one, since like we usually do 2 of them. Take a look at this guy and move through this. Now, 1A or 1B?
1B.
Yeah,
1B.
clear. Yeah, super clear. And let's go to meaning we go to 3. Body has in 3, A or B? Which one is it?
Without apparent arms, so it would be B.
Correct. It does not have clear 5 arms, right? So it is going to be B. So there we move to 5. Animal is soft-bodied and furry. Animal is not soft-bodied. What do you think?
A.
This is soft-bodied, yeah.
Yeah. We mentioned it is soft bodied. And by the way, yeah, sea cucumbers are very soft, right? I mean, you can actually touch it. You can like sort of press on it. It's very
They look soft, so.
you're 100% right. It looks soft, and here actually I mentioned it is soft bodied, right? And it is actually soft. Those are things that looks like spines. They are not spines, right? You can actually like poke on it.
They
And
look like jackfruit, if you know
ha!
what. Yeah, kind of the similar.
Yeah, actually, so who said that? I couldn't see. Was it Monica?
Not me.
Yeah.
Sorry.
Yeah. It's funny that you mentioned jackfruit actually. How many of you know what jackfruit is?
Do.
Yeah, sort of. Yeah, jackfruit is actually kind of a it's a now depending on the species, some jackfruits could be actually as like twice larger than my head. Right? Oh, it's like I have seen jackfruits that are like about 4 times the size of my head. Some of them are very small, right? Quite small. But yeah, like morphologically, they look like a jackfruit. And jackfruits are actually very, sturdy. Mm. It's a tropical fruit that actually grows in multiple tropical regions, largely in South Asia and Southeastern Asia, right?
The Caribbean is a lot like in the Caribbean.
Yeah, you know what? I always wondered whether it was introduced to Caribbean islands.
Yeah, I think it came from Asia with
Yeah.
the seed and stuff.
Yeah. Reason is it's a Caribbean islands are also tropical, so they do really well. I don't think they are invasive, but they're doing really fine in Caribbean islands. Do you
Yeah,
guys
no,
know?
I don't know. They aren't invasive. They just like
Yeah,
just grow and
yeah,
that's it.
yeah. They're edible fruits. Like I don't know how popular it is in Caribbean islands. Do people
It's
eat it?
very, very popular.
Really?
Yeah, I
They
like it
have like
actually.
festivals for it too. Like
Oh,
like
really?
ice cream. Like they have a whole thing where they make ice cream, like fry it, kind of like
Yeah.
curry. They have curry festivals and all
Oh
that.
yeah, I mean, we South Asians are not that creative when it comes to food. We all eat it in the same way. We probably should
Put
learn
it
from.
on everything basically. You can boil it, you can fry it. If you can
Oh yeah,
fry it,
yeah. And actually, people even eat the seed. Like I mean, I have heard sometimes, like it,
yeah,
yeah, like you know.
you dry it and then you roast it or you boil it and
Oh
then
yeah, both actually. I like both ways, by the way. And alrighty. And people ask always when I talk about jackfruit, is it like durian? Not exactly. They are
maybe the
completely.
outside, but
Yes, outside is, but actually the texture of the fruit, like it's more like, the jackfruit is more like tough. But durian is much more like buttery. But let's
it's
talk more about.
smelly. It's a
Yes.
strong odor.
Yeah. Yeah. Well, let's. I have a lot of fun plant stories. We'll reserve that to the next half of the semester. Um, and Monique can actually contribute with her fun stories. So this is actually like I always create this particular slide with the scientific name juxtaposed with the English name, right? So these are the exact scientific and English names of the species in that particular slide, right? Particularly when you are looking at the homework, you need this to pick up what exactly the scientific name is. Now, guys, let me tell you this. If I ask for a scientific name, genus name, species epitaph, right? This is a scientific name. Like, what happens to my pointer? Genus name, scientific, the species epithet, right? Collectively makes up the scientific name. Make sure if you are typing it, italicize it. If you are writing it, underline it, right? Don't underline if you are typing it, and don't try to italize if you are writing it. I know we have someone in this group. I believe I want to say it was Randolph, who's actually a cursorial writer. That's fine. If you're a cursorial writer, don't change your handwriting, right? Just underline it, right? Um, by the way, like, I can count the number of undergrads who actually have cursorial writing. So I get excited when I see one.
You have to
Um,
capitalize the genus name also.
Brilliant point. Excellent question. The genus name must be capitalized. Everything else, lowercase. Excellent question. Oh, one more thing. What if you don't know the species epithet? Like in Dugesia, do you know what to do?
SP.
SP, right? Brachiostoma space SP and a period at the end, right? So that's actually, and SP should also be capitalized. When you do research, you might find some people writing SPP. Don't trouble yourself with what that means. How different that is from SP. There is a difference. I can talk about that later. There's a standard where if there are multiple species for a particular genus, we put SPP, right? But if there's only a single species for that particular genus, we put SP. But like, don't trouble yourself with that. Like, I'm just telling you, for like, in case you find that distinction. But for you guys, if you only know the genus, don't know the exact species, genus space SP period. Now let's talk about sea star. The exercise on sea star, lancelet, and sea squirts. Remember, for those three critters, do this part like food, movement, skeleton, yada yada yada, right? So now I did explain these parts in my introductory PowerPoint, right? I explained most of these parts. Some of these parts you might have to look up. Give me a shout. Should I quickly revise some of this, or are you all set with just referring back to this the PowerPoint I just did?
You can go over
Maybe
it.
one, yeah.
Okay, more than happy. The more you ask, the more I do. So let's go for guys. Anyone, any of the three animals that you want me to talk about? Sea star, lancelet, sea squirt.
I don't know if you went over what a sea squirt was.
Oh, I will. I will. Of course. No, I probably might not have. Sea squirt is the same as Siona. Okay. It is the same as Siona. Okay, I will go on. Shall I go with the sea squirt as the example, guys? Okay. The food. Help me out. What kind of food do you? What kind of food do you think they eat? Like, what kind of a feeding strategy do they use?
Filter feeding.
Filter feeders. What structures they use for food capture? Well, always start from the very beginning, right? In-current siphon takes up the food, right? And then goes through the in-current siphon into the pharynx, right? And the food particle gets trapped in the pharynx, and then the water is pumped out of the excurrent siphon, but whatever the food particles that was trapped goes into the intestine, from intestine to stomach, from stomach to the intestine, from the intestine it goes to the anus, and the anus dumps stuff into the atrial cavity, and the atrial cavity dumps things out into the external environment, right? And we will go through that pathway when we go through the homework. I believe we might. Movement. What do you think about their movement? Think about the larvae, think about the adults, right? Larvae are free swimming, right? They actually have a tail, which includes the notochord, which helps them to generate movements, right? And there are muscles that actually help them to generate movements. And then the larvae is movable. Adults are permanently settled, right? So actually, if you wondered how the larvae settles, larvae actually settle from their anterior end first. They secrete a cement and settle like head first, and then completely undergoes transformation. And when you have an adult, they are completely anchored, settled on the substrate, right? Um, Skeleton and body structure. What kind of body structures, skeletal structures, do sea squirts have? Again, think about the larvae as well as the adult. Adults have a notochord, right? So notochord is part of their skeletal system, right? Um, and what else help maintaining body structure as an adult? Well, notochord recedes, right, entirely, or to like a very small portion. So as an adult, their body shape is maintained by two things. One, the pharynx bloats up and maintains the body structure, body shape. Secondarily, well, pharynx is very flimsy. It might bloat up and create a shape, but what maintains the actual shape? Tunic. Tunic is an external covering that you find to the outside of that particular animal, right? So as adults, the larvae has a tunic, but that tunic hardens up and becomes more rigid to maintain body shape, right? Um, And then, of course, symmetry, they are bilateral symmetrical. Cephalization, larvae show cephalization. Adults do not. Adults lose their cephalization. They have a body cavity which is called atrium, and I will show you what atrium is, right? Reproduction and development, well, I mentioned that sea squirts, urochordates, are actually, they have sexes separate, they expel sperms and eggs, external fertilization takes place, right? And then the larvae, the zygote after fertilization becomes a larvae, and the larvae undergoes subsequent development. Respiration and circulation, well, for respiration, they actually have the pharynx. For circulation, well, they do have a circulatory system, by the way, right? They have a heart, they have blood vessels that actually pump blood, right? You know, it's really interesting, like really interesting. Sea squirts, I mean, when you see subsequent images or the images that I already showed you, they are very small creatures, right? But still they actually have circulatory system. And other organs, they actually have reproductive organs. They do have a digestive system, a complete digestive system, digestive tract. Um, and they do have sensory structures. So phylum characteristics, well, again, you know, I showed you the phylum characteristics before. They are deuterostomes, they are chordates, they are urochordates, right? Now, and then let's look into the sea star anatomy a little bit more, right? And then we will subsequently also, I will go through labeling the anatomy of Siona as well. So what you're going to do is actually using pictures that I'm going to show you, you should be able to label these structures, and I will label some of them with you, right? And then you're going to watch the two videos to cover their external anatomy and the internal anatomy, right? So the parts that I'm mentioning over here as following structures, these are the stuff that I will expect you to label yourself in the practical. All right. So remember, I did cover some of these stuffs very briefly. Let me actually go really quick somewhere over here. Okay, over here. Let's cover some of these parts first in the diagram, right? So if you are looking into the aboral surface, oral surface is the surface where you have the mouth, which is on the underside of the sea star, right? In a live sea star, on the top side is actually aboral surface, right, where you have the anus, right? So this is the anus, right, and the madreporite on the side, okay, and then anus has a very short esophagus and it opens to the stomach, right? Stomach has two parts, right? Pyloric stomach and the cardiac stomach, right? And then the stomach leads into the intestine, and then and then of course should start from the other way around actually. Sorry. If you start from the oral surface, the mouth opens to a short esophagus. From the esophagus, it opens up to the stomach, right? The cardiac stomach followed by the pyloric stomach, right? When you dissect it from the anus end, right? So because of that, when you dissect it from the anal end first or from the aboral surface first, what you see is the pyloric stomach first and the cardiac stomach underneath it, right? But in reality, it is cardiac first, pyloric next, right? And then of course there's a short intestine opens up to the anus. What are the external structures we see in these guys, right? So opening to the stomach are actually these very elaborative digestive glands. Digestive glands occur primarily in the arms, and the arms dumps the digestive juices into the stomach in the central disc. So the stomach, mouth, anus, short intestine, all of those are located in the central disc, right? But the digestive glands are on the arms. Now, when I ask you to label all the systems in the digestive tract, don't forget the digestive glands, right? I noted that in some of your homework. Now, these guys can also, when they feed, they actually extrude their stomach to the external environment digestion happens externally, and these guys, unlike sea cucumbers, can take up the stomach back into their body, right? And what other parts do we see? Well, like I mentioned, as these guys mature, or when these guys reach their reproductive season, right, the entire arm, right, like entire space of the arm can be completely taken up by gonads, right? The digestive glands can recede all the way back to a tiny part, back all the way up here, at the edge of the central disc, right? And then of course you see these structures that belongs to their um, water vascular system, starting from madreporite, right? And then you have this ring canal, radial canal, and then of course you have the lateral canal, and then the tube feet. So this is again like kind of a close-up of the tube feet. Tube feet actually has a podium, right? And the ampullae, right? And this is the lateral canal, right? This tiny part is the lateral canal, and this particular part is the radial canal, a cross section. This is if you actually cut open a tube feet as a cross section, if you look at it. And this is actually kind of showing you the same, much more elaborative picture, right? You see the tube feet over here, see the radial canal, lateral canal, right? And the gonads, right? Over there. And the pyloric CK is another word, another term for digestive glands. It's nothing different, the same thing, right? And again, interestingly enough, now you can see the ossicles, right? That is sort of all the way surrounding the entire body, right? All right. And again, the structure that I cannot show over here is the stone canal, which actually connects the madreporite to the ring canal. So the water movement is madreporite, stone canal, stone canal to ring canal to radial canal into lateral canal into ampullae of the tube feet, from the ampullae of the tube feet to the podium of the tube feet. Okay, that is basically how that happens, right? So the water vascular system, can exchange water with the external environment as they want. Okay, let's go back to our exercise. Now, I show you the basic anatomy. Now let's work with me. Surface of the sea star away from the opening to the digestive system. So where you can see this particular calcified opening, right, that goes into the water vascular system. What surface is this? Oral or aboral?
Aboral.
Aboral, that's correct, right? And this central area, not a point, the central area of the sea star is called what? Guys? Something disc. I heard. Kenny, go ahead again.
I said, is it central disc?
Central disc, that is correct. And what do you call this calcified calcium carbonate rigid structure that opens up to the water vascular system, starting with M?
Madreporite.
Madreporite, Randolph, correct. Now let's look at this structure. So what we have done over here is flip the thing over, right? So the surface you are looking at is what now? Now you are looking at the underside. So oral or aboral?
Oral surface.
That is correct. That's the oral surface. Now, these thousands of structures you see that actually help the sea stars to move. There's lots of teeny things. What are those? That can be extended when they are filled with water.
The tube feet.
They are to the tube feet, correct. Now, in the lab handout, it asks you to count the number of tube feet. Don't try to do it over here, right? It's impossible to do that on this picture. It could be like on the order of hundreds, in certain cases thousands. So don't even try to do it. Areas in which these structures are found. Tube feet are found in a particular structure called ambulacral groove, right? The label diagram actually has that spelling, and the lab manual also has that spelling. and then hard calcified structures for protection, right? So what are these hard, externally visible calcified structures that that could offer protection?
Spines.
The spines, correct. Now I said externally visible, right? If I didn't say that, you could say ossicles. It would be correct because I didn't say externally visible, right? So spines is the correct answer I was looking for, right? And this centrally located opening that goes into digestive system is
Mouth.
mouth, correct, right? And now over here, what we have done is you look at the sea urchin, scrape the surface, right? And took that scrapings into a light microscope and you are looking at it, right? So, and what you first see is this much larger, hard, bony projections, right? And those are what? They could be not just microscopic, some of them are actually macroscopic. They project, they could be sharp.
Is it the jaw?
We actually just said what this was earlier. It's a spine, right? So the spines that are actually, when you took a scraping, the larger spine does not come off because they are much larger. Smaller spines actually come out, right? This kind of spiny structures that are bluntly ended, that are harder, are actually spines. Now, what are these? They have two jaws, so they could be movable. What are those? Those are pedicellaria, right? Remember, those actually have a stalk. They are on the surface and they have muscles to move the two jaws, right? Now, the purpose of those pedicellaria is actually for grooming purposes, right? In fact, because of the stalk, they can actually bend over and like pick stuff up, particularly parasite like leech, not leeches, other parasites. They can actually like pick them off and throw them away, right? And in fact, there are actually marine ectoparasites that these guys actually, pick out. And lot of tube-like projections on this flimsy tube-like projections on the skin that is used for respiration. Those are
Like bronchies?
correct. Dermal branchiae, right? I have read the word dermal papillae as well, but I prefer dermal branchiae better. Internal anatomy, we actually went through this already. Let's look at the actual anatomical features over here, like how it looks like in a dissection, right? So what do you think? So question for you guys. Should we do a Q&A with you guys just to make sure you know what these things are, or are you all set by doing it on your own with the video? Give me a shout. Yes, do it, or no, skip it.
Yes.
Okay, I have a yes. Alrighty. Unless you all say no, I will do it, right? Because one student asks, I'm going to honor that. So so the stomach, there are two parts of the stomach, and one part is closer to the mouth, one part is further away from the mouth. What part is closer to the mouth?
The cardiac stomach.
Cardiac, correct. And it is easy to remember. Cardiac, heart is actually closer to this mouth, right? So like that's how we remember that. And then the stomach part that is further away from the mouth,
The pyloric stomach.
pyloric, correct, right? And the reproductive organs that actually, is right now in this tiny little part, but which can fill up the entire space, those are called
Gonads.
gonads. Very good, guys. I want everybody else to take part. I do appreciate Kayla and Emily answering questions. And and these glands that actually secrete digestive enzymes, what are those called? The word is actually, answer is right there. Yep,
Someone wrote it in the chat.
yep, digestive glands. Hey, Gerard, you know, like, thank you for responding. Right, I mean, if possible, I would like you to actually say it out loud. Yeah, that's correct. That is digestive glands. So, and then, you know, okay, like I will not go over all of this, right? You know, I will ask you to, you know, watch the video and actually label them on your own, right? Um, again, here this is actually labeling of the, what is that? Oh, I see. No worries, man. So this is actually the part of the water vascular system. Again, like, you know, towards the end, if you want me to go back and, you know, like help you out, I'm happy to, but right now I will skip them because these are straightforward. That's why I'm skipping them, right? We did this already. So here, like what I have done again is on top of the video, I actually have like provided additional label diagrams for you guys, right? With external and internal. So again, you know, they're very self-explanatory. Things that I might consider explaining. So again, there's another term. Hepatic cecum is the same as digestive gland, right? So they are not something else. Hepatic means like liver, right? Cecum is like glands. So again, those are nothing new. Those are actually glands that act like a liver, right? They actually produce digestive juices. All of those words are correct. I'm not expecting you to know all of those words, but if you provided any of these words as a correct answer, I'm more than happy. But simpler the better. So in a multiple choice questions, I may not include hepatic cecum. I might include digestive glands to be simple, right? I'm providing all the verbatim for your knowledge. Now, so again, there are different sections. This is an arm cross section, right? You can see these glandular parts are actually the digestive glands, and then gonads are like more like solid fills, right? They are more granular, not glandular, right? Granular means you see like lots of like, you know, fill ins inside of them, right? Those are the same. Yeah, let's actually now move into the lancelet, right? Or branchioxus, right? So this is the anterior end, right? Posterior end, dorsal, ventral, right? It's easy to understand those orientation. It's extremely difficult to orient a urocoded adult because they undergo like an axis transformation, right? So we are not covering that, but here I do want you to know the orientation. On the anterior end, the sort of the snout-like part that projects is called rostrum, and then the stiff rod that extends the entire length of the body, right, is actually the notochord. And then in the mouth region, you actually see lots of tentacles, right? Collectively, all the tentacles, all those projections make up an organ called buccal cirri, also known as the wheel organ. I like the word wheel organ because it looks like with this multiple cirri, it looks like a wheel, right? Which actually essentially creates a water current to draw water in and helps filter feeding, right? So and then yeah, that's why actually so wheel organ versus buccal cirri, like are they different? Well, rather they are co-located in the same area inside the buccal cavity. So rather than using them as two different organ systems, I would rather consider them as a one system that helps filter feeding. Followed by, it opens up to this large pharynx with lots of pharyngeal gill slits, and then what happens is that actually the pharynx connects to the stomach, which gets into the intestine, and intestine empties through the anus, right? Now, what happens to water that filtered out from the pharynx? It gets through the pharyngeal gill slits. From the pharyngeal gill slits, it gets into the atrium or atrial cavity, which is a space between the outer tunic and the pharynx, right? And from the atrial cavity, that water goes out from the atriopore or atrial pore, right? So there are two openings, right? Three openings: the mouth, anus, which actually expels poop, and then the atriopore that expels the water that has been filtered out from the pharynx, right? It's extremely important to understand that water flow and the food flow separately, right? um and then of course they actually have a tail, and there is a caudal fin on the tail, and on the dorsal side there is also a fin called dorsal fin. These guys can move. If there's a movement, there should be a muscle, right? And those muscles are called myomeres, which you find over here, right? And you can say, you can kind of see actually myomeres are right on the top of the notochord. That also makes sense. If there are muscles that are myomeres, those muscles should attach to something. So they are actually attached to the notochord. Again, the same diagram, different diagram with the same labeling. And now let's move into Siona, right? Which is also known as the sea squirt. So now let's look at actually, a colony looks like this, and a single individual, is sort of like a chimney with two of these openings, right? Incurrent and excurrent siphons. In a preserved specimen, it is extremely hard to tell what is the incurrent siphon, what is the excurrent siphon. It is very hard to tell, right? So remember the previous urochordate we talked about, the cephalachordate we talked about, the Amphioxus had a mouth and an atriopore, right? Mouth took water in, atriopore expelled filtered water out. These guys don't have a mouth. Instead, the mouth has become the incurrent or the oral siphon, right? Don't necessarily have an atriopore. Atriopore has become the excurrent or atrial siphon, right? Incurrent, water goes in. Excurrent, water expelled out, right? Oral, mouth-like siphon. Atrial siphon, the atriopore-like siphon, which expels things out, right? Again, two different terms. Oral siphon is the same as incurrent siphon. Atrial siphon is the same as the excurrent siphon. Okay. And then if you look at this label diagram, you can see the entire body of the Siona is occupied by the pharynx, which is, named a brachial sac. Why different name? Because the pharynx has enlarged, right, occupying the entire body volume, right, and become a sac-like organs. That's why it's called a brachial sac. Now, is it incorrect to label that as pharynx? No, right? Brachial sac is a term I want to introduce because to highlight the fact that the pharynx enlarges when they transform from larvae to the adult, right? The test is the same as the tunic, right? Tunic is the outer body covering that sort of protects the entire organism. And right at the inside of the oral siphon, you get actually these tentacles that creates a water current that drags water inside. In fact, in a live animal, the siphons can pulsate, right? Meaning they have muscles, right? So how pulsation is a movement, right? So there should be something actually doing the pulsation. Those are the muscles. Now on one side of, like, sort of on the one surface of the brachial sac, aka the pharynx, there is something called endostyle. Now endostyle is actually the surface that actually produces a lot of mucus. Think about the point of that mucus. Think about what the endostyle does. If this guy is doing filter feeding, there should be some mechanism to actually retain trapped food particles. So endostyle kind of helps to trap that food particle and channels that trapped food particle towards the stomach. or rather towards the esophagus, a short esophagus that actually ends up in the stomach, right? And that goes to an intestine, and intestine ends in an anus. Anus empties its stuff into the atrial cavity. The atrial cavity, or the atrium, is the space between the brachial sac and the test or the tunic, right? And then from that atrial cavity, things gets expelled from here, from the excurrent siphon, right? Again, I'm not going to go over, right? This is the same diagram. And remember what I mentioned about the nervous system they have, right? They actually do have this, like this super bright white stuff that you see over here. That's actually a central ganglion. If they have a central ganglion, this is actually telltales of a central nervous system, right? That actually has really well coordinated functions, right? And of course, in a well preserved specimen, you can see muscles, longitudinal muscles that go lengthwise, and circular muscles. So that's actually what helps these animals to contract and pulsate to generate a water current, right? And of course they have the reproductive system, so you can see many other structures in a well preserved specimen. In an actual specimen that you see, you may not, in a museum specimen, things might be not that clear, but this is a very, very well preserved specimen, right? Now, tunic that covers the entire body, or the test that covers the entire body, at the bottom has created an anchor that helps to anchor the animal to a substrate, right? What is not shown over here, probably it was shown over there, not only over here either, is that they actually have a circulatory system. Oh, look at this, the heart. So they actually have a circulatory system, right? Right, that circulatory organ over here is not visible, is not marked, not very clear. And the stomach, you can see, is pretty large, right? And since the pharynx, or the brachial sac, or the brachial basket, occupies the entire space of the animal, right? Most of the body organs, the visceral organs, are kind of pushed towards the bottom. That's why you see the reproductive system, the stomach, the heart, and all of that towards the bottom of that animal. It's kind of the brachial sac, or the expanding, developing pharynx, pushes all the other organs towards the bottom or towards the side, right? Okay, now after that, it's simply a display and followed by answering questions. So this is actually, again, labels, right, of the Amphioxus that I'm showing you over here. So I just do want to point out that the very, very thin, flimsy dotted line is the nerve cord, and notochord is below that. So the notochord is actually much thicker. Nerve cord is very, very smaller and very tiny, right? In real life too, the nerve cord is much more smaller, notochord is much more larger, right? And this is actually the dorsal fin that actually helps the animal to move. And nerve cord, the notochord extends all the way to the rostrum. Nerve cord does not go all the way to the rostrum. Okay, that's another thing. So notochord is longer than the nerve cord. That's another way to remember it. All right. Again, what is atrium? Atrium is a chamber. It's kind of a space that occurs between the outer body covering and the pharynx, right? Again, those different labeling, we already went over these parts. So this is actually how an actual preserved specimen in a museum looks like, right? You have this incurrent and excurrent siphons. Usually excurrent siphon is larger than the incurrent siphon. Usually the incurrent siphon or the buccal siphon is larger than the excurrent or the atrial siphon. The reason is you need to take a lot of food in and then allow time, or increase the residential time inside the pharynx to filter the food, to retain the food, right? That's why actually those two are different sized. But some specimens, extremely hard to tell what is incurrent, what is excurrent, right? In that case, completely okay to label them as incurrent and excurrent siphons. But if you see completely two different sizes, larger one is incurrent, smaller one is excurrent, right? And then these sort of horizontal bars that you can see, those are actually pharyngeal gill bars or gill slits, right? And then this slender tube-like structure is actually the intestine. The round structure is the stomach, right? And even in live specimens as well as in preserved specimens, this intestine and stomach has food. So it appears in like a solid form. Well, that's actually all I have. And all we have to do is go for the homework assignment. But before that, any questions regarding any of the things we covered so far? Very well. Let's go over the homework. Again, just give me a nod. Both of you, all of you see my homework sheet? Very good, thank you. Let's go on and work with me to fill this in. So, sea star, sea squid, lancelet, right? You know the phylum, right? Sea star, Echinodermata, sea squid. Remember there's a phylum and a subphylum. Phylum is Chordata for lancet. What is this sea squid subphylum, guys? Give me a shout.
Eurochordata.
That is 100% correct. And lancelet is? What is the subphylum?
Cephalochordata.
Correct. A way to remember this is lancelet has cephalization, has a head-like area in adult and larvae. So cephalochordata, chordates with a head. Urochordata, well, whatever the left, right? That's actually how you could remember it. Moving forward, feeding. Okay, everybody, start answering me. You can turn off your mics. Sea star, how do they feed? What are they?
Predatory.
They are predatory. Very good. Sea squirts, how do they feed?
Filter feed?
Sorry?
Do they filter feed?
Filter feeders, that's correct. Lancelot, how do lancelets feed? What do? They are filter feeders as well, right? They are also filter feeders. Now, here's food for thought, right? Okay, sea squirts, particularly Siona, they are filter feeders, 100% correct. Are all Urochordates filter feeders? What do you guys think? Anyone wants to take a stab? Guys, feel free to make mistakes. If you make a mistake, I can actually correct you and tell you why we are wrong, right? What do you guys think? Yeah, that is correct. That is correct. They are not all cephalochordates are filter feeders. There are actually predatory cephalochordates, right? In my lecture videos, actually, I have some of you are in my lecture video. Some of you have access to my lecture video, but those who does not, like, I mean, you have access to my YouTube channel, so you can still link to those, check those out. There are predatory urochordates, right? That can eat vertebrates for food, right? That's how cool they are. So now when I ask for feeding type, do not mention things like omnivores or carnivores, right? It's what they eat, right? If I ask what they eat, sure, omnivore is an answer that I could accept, but here we are asking for how they eat, right? So it should be the predatory, filter feeders, scavengers, detritivores, things like that. Structures and organs used for digestion, like starfish. Can someone start naming structures and organs? Always starts with mouth.
Mouth.
Mouth. And let's go slowly down the road. Mouth,
Esophagus.
Yeah, they have a very short esophagus, and sometimes, in certain anatomical labels, they don't even label them. So, and that short, very short esophagus opens up to,
Stomach.
yeah,
Mm.
that, and
Yes.
also, you can say stomachs or cardiac and pyloric stomach, right? And then anus. Don't forget
Digestive glands.
digestive glands, also known as pyloric CK, also known as hepatic CK. Again, digestive glands, more than enough. Let's do the same for sea squirts, right? Starts with, now think about these guys, from the adult form, right? Starts with what? How do they take food in?
Incurrent siphon.
Very good. Incurrent siphon. That goes to what? The large thing.
The pharynx.
Yes, the pharynx, pharyngeal basket. I gave you 1,000 names. I'm not going to mention all of them again. And then from that pharynx, it goes to what?
Stomachs.
Before stomach,
Oh, no.
esophagus, right? Esophagus, stomach, intestine, right? And anus. Um, how about lancelet? How would you label that?
It's like a tube.
Mm,
No.
they also have a mouth, right? Mouth. Right after mouth, what do they have? Things with a lot of tentacles?
Wheel organ.
Wheel organ or buccal cirri. I will accept both of them, right? And then it goes to pharynx, and pharynx goes to what?
The anus.
Pharynx does not directly go to anus. Like, that's actually something I can tell you without a doubt. I'm glad you said that. Usually pharynx to anus, we cannot think about any animal. There are about 2-3 organs in between, right? Esophagus, intestine, stomach, sorry, esophagus, stomach, intestine, anus, right? And then, all right, locomotion. Structures used for locomotion. Tell me, you guys, sea star, how do they move?
The tube feet.
Perfect. What else do they require for movements? Tube feet are located in what? The arm, okay? Yeah, so arms are important because arms are somewhat flexible. Sea squirts, do what can you tell about their movement? Now think about the larvae versus adult, right? Because it's so dramatic. Larvae can swim, right? They have a tail and with the notochord, and adults are attached,
So
right? Somebody
the
was looking.
S word. I don't know. This or something. Or would that just be like lifestyle?
Sedentary, I think. Oh, sessile. I think that's what you're looking for.
Yeah.
Yeah, I think it's either or that you're looking for. Um, so you're right, and then actually, yeah. So you're right. They are sessile, or they are sessile is a life form, right? They are sessile meaning they're attached, right? Or sedentary meaning they are, staying in one place. And for them, for that particular purpose, they have a hold fast. That's what you call, right? It's a part of the tunic, part of the test that actually cements itself onto the substrate, right? Lancelets, how do you describe their movements, attachment structures? Now that's all a little complicated because they use their tail, right? And to burrow onto the substrate and attach, right? And they use the tail and the fins and the muscles to swim, right? So you have to think about all of those forms. Let's talk about structures for maintaining their shape. Now, guys, start talking with me, everybody. See, stop. How do they actually, what kind of a skeletal system or a support system do they have?
Exoskeleton.
Well, do any of the echinoderms have an exoskeleton? They do not, right? All echinoderms have an endoskeleton, right? So you can say endoskeleton, you can, and you can also think endoskeleton is made out of what? Remember?
They use water, but I can't think of the name of the system.
Now, I think, was it what, was it Kayla? Kayla, that was actually water vascular system is actually not what I'm referring to. Endoskeleton. So water vascular system is inside the endoskeleton, right? Endoskeleton is made out of other bony structures called ossicles or bony plates.
With the central disc, or would that just be like?
That's a good question. Central disc is actually the center of the animal. There are inside the central disc, do you find skeletal system parts? Yes. Do you find part of the water vascular system? Yes. Do you find part of the organs? Yes. So the entire body of the sea star has two parts, or the entire body of many echinoderms, including ophiuroids, have two parts: radiating arms and the central disc, right? So central disc is not necessarily something that helps them to maintain the body shape, right? There are parts inside the central disc that actually help them to maintain the body shape, right? Um, is that clear, guys? It's really important that you know the differences between the water vascular system, the endoskeleton, and the other parts. Like if you are not clear, please shout at me. Okay. Okay. Sea squirt. Now remember, the larval sea squirt had the notochord for support. Adults, how do you think they maintain their support? What kind of a structure maintains their body form, body shape? So think about what I mentioned, what you find externally in that entire body, the tunic or the test, right? That's what actually maintains their body shape. Also, this is really interesting. What they have gained is with the enlargement of the pharynx, with the ability to fill water into the pharynx and into the atrial cavity, they have regained hydrostatic function. Not necessarily with a coelom, right? In these guys, all of these guys, coelom has been extremely reduced, right? What they have is with the tunic and the pharynx, they have regained a hydrostatic function, right? So hydrostatic, you can call hydrostatic skeleton as a organ system that helps sea squirts to maintain the body shape, and also the tunic or the test also helps to maintain the body shape. Lancelet, what helps them to maintain their body shape? Now, for adults and larvae, they actually have one big advantage, which is what?
Mm,
Yeah, go on, Monique.
I just had it, but I don't think.
No, go. It's okay. Like, I mean, making a mistake, don't be shy.
I'll
But
pass this time.
sorry.
I'll pass this
Oh,
time.
you'll pass this time. Anyone else want to take a stab?
Would
Emily?
the notochord be considered?
Yes, not a chord. Correct. Moving on. You all know that all of them are triploblastic, and you already know that they all are deuterostomic. Type of body cavity. Now, here's a question. I'm not asking for the type of the actual name of the body cavity, right? Just tell me, do they have a true body cavity, regardless of the name you give it?
Yes.
Yes, all of them have a true body cavity, right? So the answer, the type is actually different, right? The actual body cavity for sea squirt and the lancelet is the atrium, right? Um, and for the sea star, they actually have the coelom, right? So yeah, they have a true body cavity, right? So they do. So So that's that. And then reproductive organs, remember, like in sea star, we saw gonads, right? And then in sea squirts, right, I did mention, testes and ovaries, right? In testes and ovaries in the label diagrams. So if you go back and take a look at those label diagrams, I actually labeled testes and ovaries in the same animal, meaning urochordates, right, as well as cephalochordates, right? No, sorry, I take it back. Urochordates, unlike cephalochordates, are hermaphrodites. They have both male and female systems in the same individual. But in lancelet, right, they are either male individuals or female individuals. So males, sexes are separate in cephalochordates, sexes are together in urochordates, right? Fertilization in all of these are external. We already established that, right? Because I mean, it's really tricky to think about fertilization, but I can give you a very good rule of thumb. Any instant where you do not observe copulatory organs. Copulatory organ is like some sort of a structure that helps the males to deliver sperms into the female system. Whenever there is a copulatory organ, right, in the male arm, you could be looking at internal fertilization. If you don't see any structure that supports sperm delivery, anytime you don't see a particular male organ that delivers sperms, you are looking at external fertilization. Here's a question. Are there any animals that actually have a sperm delivery system or a sperm delivery organ, or but fertilization is external? Are there any animal like that? Anyone wants to make a guess?
I think so, but I'm not sure.
Yes? Annie, you won't. You're right. There are. Annie or anyone wants to make a guess?
Are they land or sea?
Oh, good question. That's an excellent question, by the way. Mostly instances where you actually have external fertilization, mostly they're going to be also aquatic animals, right? So what do you guys think? Any examples that you can think of? This might be a little hard for you guys. So remember when we talk about lobsters, shrimps, many crustaceans, right, especially shrimp-like or lobster-like, crayfish-like crustaceans, they have modified swimmerets in males that help deliver sperms to the female system, right? At the same time, if you think about oligochaetes, right, they actually have mechanism to copulate with another female, right? I know oligochaetes are actually rather terrestrial, but they live in very moist terrestrial environments. In all of those cases, they have some mechanism to copulate. In terms of crayfish, lobsters, and shrimps, they have organs for copulation, but there are many well-known species of shrimps, lobsters, and crayfish that undergo external fertilization, as well as a few that undergo internal fertilization. It's the reason is, this is really interesting. The reason is, in certain crustaceans, they have mechanisms where the female collects eggs but do not permit fertilization. Fertilization happens after they lay the eggs. It's really weird. Why the hell they do that? Right? It's a big mystery. So because of this very complex nature of sperm storage and egg storage, sometimes fertilization in crustaceans happens internally in the female body, but sometimes happens externally. So that's actually why it's so complicated, right? I mean, there are studies that actually done in the last decade or last half the decade that Until then, we believed, oh, that's external fertilization. But then we figured, no, it happens internally, or other way around. So crustaceans are the weirdest creatures that I can think of, right? That actually do the weirdest stuff, right? And then because of that, in your homework, I did not take points. I actually gave award free points for the question on fertilization, right? So I think the example was the common crayfish, which actually undergoes the crayfish you studied, Procrambus, that actually they undergo internal fertilization. But if you search literature, you will get both answers. So I actually I forgot I forgot to tell you this story when I told this, but I'm glad I did, because now I can tell you the story. I read a lot of literature, right? From 1950s, 60s, all the way up to the early 2000s, right? Earlier, external fertilization, external fertilization, external fertilization. And then recent literature that's actually studied, the fertilization in vivo, they actually figured it's internal. So because externally it cannot happen according to the way that happen, according to the way these systems work. So it's pretty cool when you read literature. So okay, let's move on to that. That's kind of a cool fact thing. Now again, I'm not going to go back how water flows in the vascular system. We talked about that quite a lot. What kind of a circulatory system would you expect in the sea squirt? Open or closed? Now remember, we are in chordates, okay? So chordates tend to mostly have open or closed systems.
Close.
Yeah,
Oh,
you're right. You all are right? So actually, Siona, yeah, closed. And then Lancelet, that is something actually I did a lot of research on. Most recent literature suggests it's a closed system. So for Lancelet, again, mention it as closed, right? And circulatory organs. What kind of circulatory organs can you find in either of the there is no specific circulatory system in sea star, right? No, because their water vascular system can deliver stuff, right? And their coelom can deliver stuff, right? So they don't have a circulatory system, right? But when it comes to sea squirts and lancelets, they actually have a circulatory system. What organs would you find in? What organ is common to both of them? What do you think? So let me help you out like this, right? A circulatory system, forget the sea squirts, forget the lanceolus. Look, just think about an animal with a circulatory system. It could be closed, it could be open. Just tell me organs that you would expect to find in a circulatory system.
Heart.
Heart. I mean, why do you need a heart? Well, because you need something to pump blood, right? Again, you can think like go a little bit backwards. Okay, if there's a circulatory system, there should be a fluid. It could be blood, it could be hemocoel, it could be hemolymph, some sort of a fluid, right? And then if there's a fluid, it should be pumped. So there should be a true heart or a pseudo heart or aortic arches, right? So some sort of an organ to pump. So you are right. So Kayla, you are right. Well, what else do you need?
If it's closed, blood vessels.
Correct. Even if it is not closed, right? You need to deliver from the heart to the open blood space, right? So there should be some sort of a blood vessel, right? Those vessels could have different names, right? I'm not asking for the different names of the vessels right now. I'm asking for the organ. So vessels and hearts. Now, sea squirts actually have a heart and a vessel. Fun fact, lancelets, they actually do not have a heart, right? So since they are very small organs, right, very small organisms, the pressure created by pulsating blood vessels itself can actually circulate, right? And then their organs are so small, diffusion from the blood vessels is more than enough to circulate. There is no need to maintain pressure or pumping, right? Since their systems are so small. right? They're so simple organisms, right? Diffusion helps. They are very flat and small, so no need for a heart. So the pulsation of the blood vessel itself is good enough. So no heart for lancelet. Now, guys, let's look at the comparison. Let's go over here, right? Look at the comparison. Now here, guys, pay very close attention. No, read the question carefully, right? Compare and contrast the movements of echinoderms with fused ossicles and those with separate ossicles, right? List how they move and how this affect where they live and what they eat. Give at least one example of a species with fused ossicles and one example without. Okay, now let's start with the last part. Can someone tell me, scream out an example with fused ossicles?
Sea urchin.
Very good. Okay, can someone tell me another example for a fused ossicle example?
The sand dollar.
Perfect. Great. Now fused and separate ossicles. So separate ossicles could be separate hinged, separate overlapped. Can someone tell me examples for those?
Starfish.
Excellent. Another example.
Did someone say urchin already?
Did someone say urchin already? Yes, for urchin is a fused one, right? I'm asking for unfused, non-fused.
Right. Okay.
Yeah,
Okay. I'm
yeah.
kind of in
So
the clouds.
yeah, no problem, no problem, right? Anyone want to? Yeah, James.
And I guess I'll try to say something again. Sea squirt.
So now we are looking at echinoderms only. Okay, sea squirts are chordates.
Okay.
Okay, which now here's how to think about that, right? We are looking at echinoderms. What kind of an echinoderm will you expect to have unfused ossicles? Well, an animal that have a greater flexibility in their body, maybe a greater flexibility in their arms. Can you guys think about, other than sea star, what other echinoderms have the greatest flexibility in their arms?
What's the skin? Is it the octopus? I don't know. Yeah.
The one that looks like an octopus,
Looks like
but not an
octopus. Yeah.
octopus. Yeah, one that looks like an octopus. What is it?
Uh,
It's a star, something star.
Brittle star.
Brittle star, very good. Okay, now let's fill in the Venn diagram. Okay, so first, I think it's easy to start with what is common. I think that's the best way to start with. So tell me how do both of them move? For the movement, do those two examples you listed, are there anything common to them that helps movement?
you know, little things.
Say what, Monica? Now? Didn't hear you quite well, Monica.
Both have like the kind of like tube-like
Yes, yes. What do you call that?
tube.
Tube feet. Tube feet. Okay. So that's actually one. How do they move? The common, the intersection of the two sides, that's going to be tube feet. Well, again, what is common to both of them in terms of where do they live? So think about where do they live. Now, don't say marine because yeah, all of them are going to be marine, right? So in a marine environment, where do they specifically live?
On rocky surfaces.
Perfect. You can say hard ocean bottom, rocky surfaces. Perfect, right? And then let's move to the separate ossicle side. Unique to the separate ossicle side, right? How do these guys move when they have separate ossicles? Remember what I mentioned, if the ossicles are separate, the endoskeleton is highly flexible.
They use their arms.
Perfect. Perfect. They actually can use their arms, flexible arms to move around, right? It's tube feet and arm that they use to move, right? What do you think? Will you think they can move? Do they move slowly or do they move really fast?
Slowly.
I was fast because they have
Correct. They move really, really fast. In fact, among echinoderms, brittle stars can move extremely fast. They can disappear right in front of you, right? Um, so on the flip side, right, on fused ossicles, well, I have to be very careful here. Like, it's easy to say, yeah, their ossicles are completely fused. So either they don't have arms, right? Like, for example, if you look at the sea urchin, sand dollars, they don't have arms to move, right? So don't say they don't move. They do move with tube feet. Um, so you can directly mention no arms. And other than tube feet, tell me what else can they use for movements?
Can they use the siphons, or are they mostly CS? Like, can't pronounce the word, but
So remember, siphons were not present in echinoderms, right? Siphons were a chordate feature, right? So these guys don't have siphons, okay? That's a good thing, like they don't have siphons. See, like whenever you make a mistake, it becomes a learning moment. Well, what I kind of didn't tell you, I intentionally saved it for this particular moment. Remember the sea urchin spines? Those spines are actually like, they are not permanently anchored onto their skeleton. They actually lie on sockets of their endoskeleton, right? Because of that, the spines can be moved, and spines actually have muscular attachment on the base. So they can actually move with their spines, right? Where So going back to the separate ossicles case, where do they live and what do they eat? So first, where do they live? What kind of a marine environment would you expect them to live?
Rocky intertidal zones.
Yeah, actually, that's an excellent. That's not a bad answer. That's actually, by the way, a correct answer, right? They can live on, in fact, different types of substrates. Some of them can live on like ophiuroids, for example, the brittle star. They can live on sediments, sandy substrates. Some of them can live on rocky substrates, right? So you can actually, you can mention, like a wide variety of bottom substrate is the answer. But can you remember what I mentioned regarding, examples? You gave me two different, you did mention of the brittle stars as the example for separate ossicles, or the basket star is another example. Can you tell me another example where you suspect that this an echinoderm where you expect high mobility in their arms? So now you have to think about an echinoderm that has arms, right? So what kind of an example that you can think about here? Go one by one, right? Definitely, urchin sand dollars, they're out. Okay. Um, sea urchins, they're out. They don't have large ossicles. Sometimes ossicles are absent in them, right? So that only leaves sea stars, brittle stars, and sea lilies, right?
I guess brittle stars kind of look fast because of how like long and skinny they are. They look like you can skitter around pretty quickly.
Yeah, right. We already mentioned them, right? We already
Oh,
mentioned
okay.
brittle stars and basket stars as an example for separate ossicles, right? Um, so you're right, by the way, right? You're still right. What other example can you think about? Like sea lilies, right? Remember in the sea lilies, those branches I showed, those branches are as flexible as arms of um, brittle stars, right? Those are actually called arms, the things that radiate. Like if I were to show that PowerPoint again really quickly, here, right? These ones, like they are extremely flexible, right? So don't forget these guys when you think about separate ossicles, right? So you can mention them, think of them as an example. So their lifestyle is sedentary, right? They are also permanently attached to substrate, hard or soft, right? And then if you move to the other side, fused ossicles, right, where would you again expect animals with fused ossicles to live?
Fused ossicles, they would be siestral.
I'm not sure what you were trying to say. You said siestral?
So when they have is the ones that have the fused ossicles, they're more likely to not move around when they're adults, like they anchor themselves.
So again, remember the example that we came up with for fused ossicles. It was the sea urchin and the sand dollar, right? So yeah. Now, guys, I'm warning you. Like this is fine. I'm perfectly fine. You making mistakes, right? Do not mix up the echinoderms with the chordates, right? It's easy to make the mix because I'm combining completely two different groups together, right? It's very easy to get confused, right? And I just covered them, and then you haven't covered these parts in your lecture. How many of you give me a shout? Are you up to this level in your lecture yet? No, no, not in mine. I mean, I assume not in the other lectures as well. Definitely not. Unless I do notice that some of the students have already listened to those lectures. Terrific, but technically that's not covered. So it's easy to get confused. Please don't confuse those two, right? The Lancelot and the Siona chordates, right? Everything else for echinoderms. So coming back, those sea urchins and sand dollars, they actually, can live in either hard substrates or softer substrates. Remember, sand dollars, fused ossicles, can live on sandy substrates. Sea urchins, on the other hand, also fused ossicles, but only can live on harder substrates, right? So, and what do they eat, right? What do echinoderms, like sea urchins and sand dollars, eat?
Algae.
Algae. So because the algae or plant material, in fact, um, um, echinoderm, in fact, sea urchins are more likely to eat larger vegetation, larger plant material like macroalgae, macro seaweeds, right? So you can actually use the name grazers, right? Rather than saying they eat algae, you can say they eat algae and they are considered grazers, because no matter what they eat, is it unicellular algae, filamentous algae, um, large macroweeds, large macroalgae, like they all the time they eat by grazing. So mention they are grazers, right? Um, Going back to those with separate ossicles, right? Separate ossicles. What kind of a foraging strategy would you expect in those? We are looking at brittle stars, basket stars, and sea lilies.
They actively hunt for their food.
That's number one. They are actually predators. Again, when you say actually, the correct word for hunting for food, predators, right? Now, not to say that Emily was wrong, but there are some, another reason why I prefer predatory is actually there are some basket stars that actually are sit and wait predators, right? They can camouflage well in the seaweed, and whenever the prey swims by, right? But sea stars, on the other hand, are actually hunting for predators, right? Well, what is the feeding strategy of sea lilies?
Filter feeding.
Filter feeding, very good, excellent. The next part is actually very you can do it on your own. It's simply you look up each of these terms. What is echinodermata? Well, echino means spiny, dermata means derm, meaning skin, right? So like that, what you are going to do is look up the etiology of these terms, break it up, and explain what that means. The root of the word, break it up into two or more terms, right? And tell me what those things means, right? In Eurocode data, Euro means tail, right? Code refers to an actual code that runs the entire length of the body, motor code or nerve code, right? So yeah, like that, like completely like sort of dissect the word look at the etiology, and then explain, fill in that particular table. The last part is fun, right? So your job is to choose one group or a lower level of classification and create a new name using other characteristics of the group. So you're looking back at either Urochordates, Cephalochordates, or Echinoderms, any of those groups, right? Pick a particular organism or a group, right? It could be a species, it could be a genus, it could be a family, order, whatnot, right? Subphylum, class, and then based on their characteristics, right, which I explained to you today, right, devise your own name, right? Come up with your own name for them. So like actual group name, right? Let's say like sea lilies, right? So the actual name is Crinoidea. So class Crinoidea, and your name, before your name, like the third part is like, what are the characteristics they have, right? List all the characteristics, do some research about their characteristic, right? And then come up with a name for them, right? For example, if I were you, let's look at the sea lily group within Crinoidea, which are attached adults compared to sea feathers, right? So, so being attached is a kind of a unique feature for adults, right? So I would call them sedentaria, right? Sedent meaning attached, sedentaria meaning, like, to kind of Latinize it, right? So like that, pick a particular feature, right? That is unique to that particular, not like 100% unique to entire clade, relatively unique to that particular clade, and devise a name based on the ethmoline data, the ethmoline this thing, right? Based on this particular website that we are giving you, right? There are a couple of websites we are listing over here, right? All of them will help you to pick names. Now, did any of you, this is a fun question, any of you took Latin in high school or taking Latin now as a like an additional language? Oh, James, where? Now or high school?
Oh, I took 2 years of it in high school.
Oh, so you don't need these databases, man.
Maybe I'm a little rusty, but
Yeah. It's high school, right? Now it's a
yeah.
while.
I
So
know derm is skin though. I'm pretty
right,
sure.
right,
Derm.
right. Yeah, like I mean, so I mean, if you have that kind of has done some fun stuff in high school, actually, um, just a fun story to share. Number of colleagues in my entire career, right? I worked for 4 universities. This is my fourth job, and before that, I have worked for 3 other universities. So I've known over like what, nearly 100 colleagues. Guess how many of them actually have taken Latin?
Say maybe like 10.
One.
One.
Yeah.
Wow.
Yeah. By the way, biology colleagues, right?
Oh, okay.
Yeah, biology colleagues. I mean, I'm sure there are like, I don't have a whole lot of non-science colleagues, but that's how few. But it will be a fun activity to do. Now, am I required that you come up with Latin name? No, I don't say that anywhere in that question. If you can come up with an English name that actually goes into a really cool group name, I'm fine with that as well, right? But don't come up with things like sedentary animals. That's boring, right? Come up with a fun name, like a single, jointed name, right? That's why Latin is favored, because that helps you to merge these things. And just sharing this out of interest, since we have one Latin taker, since I did not ever take Latin in college, graduate school, or in any of the degrees I have, um, in my job, I do actually discover new species, right? That's actually one kind of my research. So I had to actually teach myself those Latin myself, taking online web-based courses, so that, because like without that, I cannot coin, a meaningful name. When I come up with new species names, particularly like, like I never have described a new genus myself, but there are several new species epithet I had to come up with. So I need to like, make a meaningful name out of them. So just to be on the lazy side as well as to be scientifically meaningful, what I do is I either convert, try to, okay, where I found this new species. Oh, in this particular area. What is the closest town's name? And Latinize the town name. So, because I'm describing a new species, not a new class or a new family. So it's harder to come up with a unique character that is unique for only that animal. So it's extremely hard to do that. For example, this is not something I did. There are many species whose epithet is variegata, right? Meaning they have like sort of a variability in their morphology, in their coloration. So we call them variegata, right? But I am too lazy. Like I look at the town name and Latinize the town name. that's
Okay.
actually, yeah. Oh, sometimes what I do is, this is a fun fact.
Mhm.
Very recently, we named a particular gecko after one of the senior scientists in our community, right? So the scientist's name is Bauer, right? Aaron Bauer. We actually name it, we name it Nemaspis, the genus name, Aaroni, right? Because Aaron is his first name, right? And previously we actually named it after another scientist, a Portuguese guy. So he's De Silva, right? So name is actually Ceratophrora, the genus name. The species epithet was De Silvae. So yeah, so D, there's no space, De Silvae, because you have to Latinize it. So a couple of fun things. Well, we are done, people. We have an extra 15 minutes. If you have any questions, you can stay behind. I will talk to you, answer your questions. Anything regarding your poster, anything regarding your poster assignment, the one that is due, any exam practical questions. By the way, before anyone asks what kind of a practical this is going to be, we are going to provide you pictures as photograph on a PowerPoint, right? And the questions are going to come through Blackboard, right? So what you have is, you have the PowerPoint open and the Blackboard open. So you create the question, go back to the PowerPoint, look at the picture or whatever the details that comes with the picture, right? And then you go back to the Blackboard, right? You can have the two screens side by side, right? Or whatever the way you do. So that's how it is going to be. It's going to be largely based on pictures. Like 99% of the questions are going to be based on pictures, diagrams, photographs. Um, um, you don't have to study your lecture notes, but if it helps to understand lectures better, of course, go ahead and study lecture notes. But we are designing the practical 100% based on what we taught you in the lab. 100% based on the pictures, diagrams that are there on the lab PowerPoints. There are two lab PowerPoints, so it could be any of the pictures that is on those lab PowerPoints, or a similar picture, right? Different picture, but very similar to what you already have seen. The organs I want you to know how to label independently are the ones that we have already mentioned that you are required to know. So yeah, that's basically how the practical is going to be. The practical is going to be on the day of, on Thursday, right? The same time, right? So you will log on as usual. The only difference is I am not going to proctor. I'm going to because that will give you anxiety. No, I'm not going to proctor. I'm not going to ask you to keep your video on. Nope, I'm going to let you work on your own, right? So yeah, that's as simple as that. So you keep the same time, right? Make sure you have the same time. Make sure that you're on a I know that some of you had trouble with internet connection. Like, please make sure that you don't get kicked out in the middle, right? We will make sure, just to make sure you don't get unlucky, I will make two options. I'll let you take the exam twice, right? Like I do for those who are in my lecture, like the same way, right? Two attempts. If you are satisfied with the first attempt, don't worry about the second attempt. If you are uncertain about the first attempt, go back for the go back. Open book. You can look at anything Any of the you can keep your lab manuals open, you can keep your homework sheets open, you can keep your lab notebooks open, right? My advice, just keep the lab notebook with you, because like the way you manage your lab notebook, if you do it properly, anything in the lecture that is important will go into the lab notebook, right? Any lab manual part, right, that is important will go into your lab notebook, right? Any labeling I do in my PowerPoints will go into the lab notebook, right? So that a very well completed lab notebook is probably all you need as an open book. But if you are uncomfortable just with that, if you want to have everything beside you, do it. I advise against Googling, right? Because I've seen incorrect answers popping up if you Google, right? So I advise strongly against Googling. Other thing you cannot do is, again, I'm not monitoring, I'm not going to act the NSA, right? Do not communicate among you guys, right? Don't text, don't share answers, like do on your own, right? And I'm going to trust you, right, that you will not communicate among students, right? Or communicate among other faculty with other faculty members, right? Regarding the exam questions, you don't have that much time, right? You have like what, 3 hours? Is 3 hours enough to complete both attempts? Honestly, you will have time to complete both attempts and probably have like extra half an hour left, right? So no, don't worry about the timing at all. You will be perfectly fine.
So this will be during the lab class that we take
Yes.
this?
Yes. Yeah. The reason we are doing that is so that you definitely you will have nothing else during that time, because we are, this class is now synchronous, right? So and we actually have it in the system as synchronous. There should be no overlap between this and that, this and anything else. So yes, it will be made available maybe like 5, 10, 15 minutes earlier. Like I usually do that, I open it up earlier, and I probably will extend, like another 15 minutes after, in case someone ran into internet trouble, right? So 11 o'clock, I might open it at 10:45, right? Supposed to be finished at 1:45. I might give you an extra 15 minutes and make it 2, but so that's an half an hour more. Now, do I even think you need it? No. Like, I mean, normally when we do it open book in lab, students finish within within the first hour. Believe me, right? But use your time. Don't rush. Maybe rush to the first attempt and then slow down to the second attempt. Whatever you want to do, it's completely up to you. So that's it, guys. That's basically all I wanted to mention. So I'm going to let you guys go, and I will hang around if anyone has any questions. If anyone wants to hang around in case others have good questions, you are welcome to. Okay, bye.
Have a good one, Professor.
Bye. Thank you.
Yeah,
Have a
good
good
one,
one.
guys. Bye-bye. All right.
Thank you very much.
Yep. Okay, Emily, it's just you and me. Any question for me?
I just have one, but it was kind of a silly question.
No such thing. No such thing.
Sea cucumbers have the ossicles that are separated, right?
That's an excellent question. So let me answer that. Yes, because some sea cucumbers, echinoids, have extremely small, microscopically small ossicles. So you can argue those as separate. But there are also a lot of other sea cucumbers who have completely lost their ossicles, right? So because of the fact that some sea cucumbers have completely gotten rid of their ossicles, it's a little challenging to consider them as separate, although technically you are right. So for examples in terms of separate ossicles, my best example are crinoids, sea lilies, sea feathers, sea stars.
Okay.
Yeah, that's actually a good question.
I think that's all I had for now.
Okay, so shall I also close the session then? Say goodbye to you?
Oh, yeah, I had one more question about the
Yeah.
the poster for next week.
Yeah, of course.
So it's just one of us submitting a paper with preliminary information?
Exactly. So, like if you look at the this document which says lab group research project, like the PDF, if you go to the second page, right, it says submission of midterm status update report. And yes, so make sure you have identified the family, right, that you are assigned to with respect to its domain, kingdom, phylum, class, order, right, that linear classification. And then you have to list genera, right, the total number of genera,
What does
and
that mean?
yeah, I will. I have good question. So list the number of genera it contains and at least one example, right? So what that means is, tell me, what family did I give you?
I believe it's Trochilidae, the hummingbirds.
The hummingbirds, yes. Um, so hummingbird family. Now, what genera? Genera means genus, right? The genera is the plural of genus, right? So tell me how many genera, right? How many genuses? Genuses is actually not grammatically correct. How many genera do you find in that particular family, right? Not how many species, how many genera, right? So you can actually just do a Google search, right? On hummingbirds, right? Hummingbirds, number of genera, something like that. Make now this is a this is a good question that you asked. You will find 10 different answers from 10 different sources. Now, that is not because some sources are unreliable or wrong. The reason is the taxonomy, right, is actually a field that constantly gets changed, right? So because of that, you might find answers that are like very close to each other, but not exactly the same. So as long as like you pick a particular source that is a reliable source, maybe it is coming from a museum website, maybe it is coming from a research lab, maybe it is coming from a zoo, or maybe it is from a journal article, you can directly list that, right, with the source, right? Don't worry about figuring out what source to use. No, you can ask me, right? For example, let's say um, you find 5 sources saying like 4 sources says 95, another said 98, another said 90, another said 93, another says 120. Yeah, 120 is very likely to be wrong, right? Another says 80. Yeah, that is also likely to be wrong, right? But if you pick any of the other, that between 90 to 99, fair bet, right? Now you might ask, like, but why? I mean, we're scientists. Why are we keep telling 100 different things? The reason is, Emily, there are many different ways to do cladistics, right? Remember when you guys designed the cladistic tree, I kept mentioning there is no right answer. The same thing here. It depends on what features you use to create the cladogram, right? Genus is a one point in the cladogram, right? So we might use, like, you might think, okay, so if the cladogram was developed using genetic data, you must have the same number of genera. No, because what genetic data are we talking about? Is it mitochondrial genome? Is it mitochondrial DNA, mitochondrial RNA? If it is nuclear DNA, what nuclear gene are we using, right? Based on all of those, still you can end up with different cladistics, right? Okay, you might ask, okay, fine. If everybody is using the same gene, you should end up with the same number of cladogram, right? No, because the statistical algorithm, when I have published, I don't know, 5 papers maybe describing over 10 species, right? 10 new species. Do I ever use the same statistical algorithm? No. I use different statistical algorithms based on, many different reasons to describe species. So even for the same genus, right? Even for the same, like what I do is I look at the genus and try to understand different species in the genus. I use different statistical algorithms, right? So like if we are looking at identifying different genus, people will use very different statistical tools, right, to separate different members of the same family into different genuses, right, to genera. So no. So and then again, there are different groups of scientists: splitters and clumpers. Splitters, well, I am being considered a splitter because I try to look into subtle differences that actually reasonable enough to argue species separation, right? So I might, when I publish, I might actually mention a greater number of species. I prefer to pick references that actually reasonable but gives the larger number of species, right? For example, in the range from 90 to 99, I might pick the 99, right? It's a personal preference. And there are others clumpers who are extremely resistant, very resistant for splitting species, right? Because they think like for a species separation, they need more than one character. For me, like, no, it's all about genetics and phylogeny, all about genetics. So there are splitters and clumpers. Both are scientists, right? But so that's why if you wondered, like you might have already seen so many different species numbers, so many different generic numbers. That's the reason. And then list 4 different features. Again, when you list 4 different features, pick unique features, right? Pick as unique features as possible, right? And then, for example, your family are hummingbirds. Pick features that are unique for all hummingbirds, not to all birds, right? So for example, I will tell you one specific feature. Hummingbirds can fly back and forth, up and down, right? That's a hummingbird feature, right? Hummingbirds actually drink nectar, or their nectar with help of their tongue, which is a tube-like, which is a tube that can split apart, right? What happens is they actually put the hummingbird as 2 splits. This is really weird. Imagine the tongue not like that, right? It's a tube in 2 parts. It goes into water as 2 parts into the nectar gland, the liquid, and in the liquid it actually fuses and makes a tube, right? So that's a hummingbird feature, right? So the long slender beak that is curving downward is a hummingbird feature. You know, like that, pick features that are unique to the family, but um, not unique to higher order, not unique to their class, right? Not unique to the entire order, unique to the family. 3 closely related families that can be used in your phylogenetic tree. Um, 3 closely related families that can be used in your phylogenetic tree. So what is your family? Like in the phylogenetic tree, right? What are the sister families, right? What are the other branches that are actually closer to the hummingbird family, right? Just mention 3 of them, right? Yeah, just 3 of them. That's all we need. That's as simple as that, right? Any questions? Any
To find the sister families, when you would just kind of look it up also.
Yeah, you look it up. So there are 2 ways. Yeah, one is look it up, like, you know, sister families of hummingbirds, right? Or I cannot pronounce properly the hummingbird family, like that family, close branches, right? Or related branches, okay? Or relatives of hummingbird family, like that. You can try different search terms. That's one way to do it. The other way to do it is actually search for hummingbird family phylogenetic tree. You know what? Let's do it. Do you have time, Emily? Like I can, I don't mind explaining this to you, but I was wondering whether you have to go to a class or something.
No, I have time.
So doesn't help very much. Okay, this is actually terrific. Oh, this is an example, not exactly what you should be doing, but let me show this. This is good enough. All right. Do you see this phylogenetic tree, Emily?
Yeah.
Yeah. So this is actually order level, not family level, right? Um, this one is actually the hummingbird order, right? So what is its, can you tell me based on this tree, what is its other closest order?
I can't read it, but it says swifts next to
Yeah,
it.
there you go. That's correct, right? So you should do the same thing for family. This is for order. So what I typed was hummingbird family, right? Phylogenetic tree. Another one I can do is I will say Trochiliformes phylogenetic tree. Because what I'm doing is now I'm looking at, I want the family, but I go a step up. I go to the order, right? Which is Trochiliformes. Trochiliformes. I don't think that's actually the correct. Nope. Go over here. Trochiliformes phylogenetic tree, right? Now, if I go to these trees, I should have, if I spend enough time, this should come up. Come on. That is actually, it's too small for you to see, right? That is actually the family level classification of hummingbirds. So what I did was, again, what you need to understand is not how to exactly get there, like how I got there, right? Um, Typed the order name, right? And phylogenetic tree, because order is the immediate above linear level to family, right? So and here you are looking at, again, Trochilidae is actually the family of hummingbird. That's the one that you are looking at, right? So here, this is a Trochilidae family. Now tell me, based on this, can you give me at least one family that is the sister family to them?
Lesbinae?
Yes. Yeah. Yeah. So again, looking here, this is the hummingbird family, right? This is a subfamily. This is a subfamily. So this is again an example, right? So how you read it up, right? Now what you are doing is not looking for subfamilies, looking for the family. Do you understand what I am asking you to do?
Yeah.
Yeah. Alrighty. Okie dokie. Yeah. So anything else I can help you with? If you, that's a good question, Emily. It's a really good question. I wish others asked that.
Well, it's been recorded, so people can see
Yeah.
it.
Yeah. Anything else?
I think that's it for now.
Okie dokie. And if you have any more questions, you can actually always ask me. All right. And I mean, feel free to ask that any question, lab question in the lecture uh, Q&A if there are no lecture questions. Most of the time, students ask their questions and then they leave. So if you wait around the last moment, last half an hour, probably you're going to have some time to ask those as well. Okay.
Okay.
Okay. I'm going to finish in the session and right. Have a good rest of the day, buddy. Bye-bye.
Thank you.